In this lecture, we will discuss about blood. In our previous lectures, we discussed about the heart and the blood vessels. Now, the blood, which is the content of these heart and blood vessels. At the end of this lecture, we hope we'll be able to understand better about the composition of blood and functions of blood. So to start with, the blood has three main functions. Number one, transport function. Number two, regulatory function. And number three, protective function. With regards to the transport function, the blood is responsible for delivering oxygen and nutrients from the lungs and from the digestive tract to all part of the body. It is also responsible for transporting carbon dioxide and metabolic waste from the cell to the sites where they are eliminated, such as kidneys or lungs. And if you remember endocrine system, all the endocrine hormones will be directly secreted into the blood, and those will be carried by the blood to the target organs. With regards to the regulatory function, it has got regulation of temperature, regulation of pH, and regulation of circulatory volume. With regards to regulation of body temperature, it plays by absorbing and distributing heat throughout the body and to the skin surface to encourage heat loss. So during the hot weather, there will be vasodilatation and blood will be diverted to the skin for heat exchange with atmospheric environment. When it comes to maintaining normal pH of the body tissue, the blood contains many proteins and blood-borne solutes which will act as buffers to prevent excessive or aberrant change in pH. With regards to the control of circulatory volume, the blood contains plasma proteins which will keep the water in the circulation and maintain the circulatory volume. Blood also has protective function. It plays a significant role in prevention of infection. The blood contains leukocytes, platelets, and immunoglobulins, which we attack against the foreign antigens. And blood contains platelets, and even if the blood vessels is punctured, the platelet will adhere to the blood vessel wall and stop the bleeding. And therefore, these three functions are important. So now, if we draw blood from a person, I hope this is a very common procedure you all have seen. Then put that blood in a test tube, put the test tube in the centrifuge, and finally we get three layers structure. The first layer is red in color, which is made of red blood cell, which is RBC, and another large layer, which is like a straw light yellow color, which is plasma, which is the fluid portion of the blood. And in between these two, we have a white, light color buffy coat. So blood is a specialized connective tissue. And because it's a connective tissue, it is made of cell and matrix. The cell could be red blood cell, white blood cell, and platelet. The fluid portion of the blood is called plasma, which contributes 55% of the total blood volume, as we can see in our previous diagram. So now I'm going to elaborate that plasma of blood. Of course, plasma is fluid. It is made of water. 90% of plasma is water. It also contains electrolytes such as sodium, potassium chloride. It contains nutrients like glucose, lipids, amino acids, and vitamins. In the plasma, there will be urea, ammonia, carbon dioxide dissolved, all the hormones, and plasma protein. The proteins present in the plasma, which are known as albumin, globulin, and fibrinogen, they are all present in the plasma. Now I want to introduce you a new terminology, which is hematocrit. Hematocrit is the percentage of blood volume that consists of RBC. As we can see from the previous diagram, like after centrifusion of the blood, what is the percentage of RBC do we get? That is usually around 47 plus or minus 5% in male, 42 plus or minus 5 in female. And newborns tend to get higher hematocrit, which range from 42 to 68%. Another vocabulary to remember, this is serum. So what is serum? Serum is plasma minus clotting factor. So if we remove clotting factors from the plasma, for example, like fibrinogen, then we will get serum. And the plasma protein, like the albumin, which we maintain the oncotic pressure of the blood, which means it will keep the fluid of the blood in the vascular compartment. Globulins will play a role as antibodies, and it will also transport lipids, ions, and copper. Fibrinogens will play a role for clotting. So I hope you understand so far. What I want you to do, I want you to stop here now. I want you to go back to the previous slide, have a look before we continue to the next part of the lecture. I hope you get a break. Now, let's take a drop of blood from a patient. We put that little drop of blood on a glass slide and we make a blood film. So I think it is a good YouTube video, and I hope you will find time to have a look how to make a blood film. And finally, once you got the blood film, and if you put it on the microscope, you will see something like this. You will see multiples of red circular cells, which might be uncountable because they are multiple. And in between them, you will see a few like blue, purple colored cells. So these red or pink circular cells, they are red blood cell or RBC or erythrocytes. And the blue, purple colored cells, they are in combination, we call them white blood cell. And small little chips in between here and there, as you can see here, they will be platelets. So if you concentrate on the WBC, here you can see different shaped WBC in our blood film. Of course, if you look at any blood film randomly, you will see red blood cells mainly, because the red blood cell will contribute about 45% of the total blood volume, and RBC is the highest component of human cell in the body. So now if we look with the electron microscope, we will see this globular or spherical leukocyte, white blood cell, donut shaped like a circular ring, but there is no empty hole, it's just dimple, biconcave structure, red blood cells, and you will see small little chips and pieces of platelets. So let's talk about RBC, which is also called erythrocytes. They are about 7 to 8 micrometer in diameter. They are biconcave because the peripheral proteins are present in the periphery, and therefore they have like a donut ring. They are not stiff, they are deformable. They are surrounded by plasma membrane. Although we say red blood cell, actually it is not a complete cell because it does not contain nucleus, it does not contain organelles in the mature RBC, and their cytoplasm contain hemoglobin. Hemoglobin means heme plus globin molecule. So let's have a look at the hemoglobin. So for the globin portion, we have 2 alpha globin change, as you can see here, with color with light brown color, 2 alpha globin, which is the amino acid chain, and 2 beta globin change here. And in between each globin change, in the center of the globin change, we have the heme component. And if you look at the heme component is made of porphyrin ring, like a ring-like structure porphyrin, and in the center we have ion. So this ion has the ability to reversibly bind to oxygen. So one porphyrin ring, which makes the heme component of the hemoglobin, can carry 1 oxygen, and therefore in 1 hemoglobin there will be 4 ion, and therefore 1 hemoglobin carries 4 oxygen molecules. The lifespan of the RBC is 120 days. And as we know, it is a biconcave structure. By making biconcave structure, it increases its surface area by 30%. And there is no nucleus, no organelles, no mitochondria in RBC. It generates its energy anaerobically. Its job is to carry oxygen. And that's a good point here, because if the RBC is consuming oxygen for its energy requirement, the oxygen it carries will not be delivered to the cell. And red blood cell will carry carbon dioxide as well. And if you look at the size of the RBC diameter is about 7.5 micrometer. Let's talk about WBC, white blood cell, in other words, leukocytes. They are spherical in shape. We have seen that. They are complete cell because they contain nucleus and organelles. They act and they function outside of the vascular compartment. They do not have function intravascularly. They have to leave the circulation for their action. And if you look at this diagram. Initially, the leukocytes, white blood cell, will be floating with the blood. And once there is a concern with infection, for example, or inflammation, the red blood cell will get attached and adhere to the endothelial membrane, capillary endothelial membrane. And finally, via the diapedesis, it will leave the circulation and it will go and act with the antibody. There are 5 types of antibodies, white blood cells in our body. The way we try to remember is never let monkey eat banana. Never stand for neutrophil, and L, let me lymphocyte, M, monkey monocyte, E, eosinophil, and B, basophil. So this is in order of their contribution to the WBC. So let's say if you look at the blood film, you will see RBCs. If you see white blood cell, the most likely to be present is neutrophil, because they are the most numerous RBC and most numerous WBC in the blood. And again, like you can classify WBC into granulocytes and agranulocytes. Granulocytes mean there are visible granules in their cytoplasm. For example, neutrophil, eosinophil, basophil, and agranulocytes, there is no visible cytoplasmic granules, and they are lymphocytes and monocytes. So in the cell component, we said we have red blood cell RBC, white blood cell leukocyte, and platelets. So now we were talking about leukocytes. And as we know, never let monkey eat banana, never neutrophil, which contributes the 50 to 70% of the total WBC. Lymphocytes, you will see here, lymphocyte about 25 to 45% of the total WBC. Monocyte, 3 to 8%, eosinophil, 2 to 4%, and basophil, 0.5 to 1%. And therefore, if we look at the blood film, it will be highly unlikely to see a basophil because it is less than 1%. It is highly likely to see neutrophils because it is like 50 to 70%. So these are the histology features or cytology features of the WBCs. So if you look at this, we have a trilobe, 1, 2, 3 trilobe nucleus, which is lying in the pink cytoplasm, which has got granules. This is very typical of neutrophil. So neutrophil is multi-lobe, but usually 3 to 5 lobes, and they are present in the nucleus are like dark purple color, which is present in the pink cytoplasm. In eosinophil, we have bilobed nucleus in the red or pink cytoplasm. So it looks like an ET. So bilobe eosinophil looks like ET in the red cytoplasm. Basophil, which is the least numerous WBC in the blood, it has a little bit characterless appearance, but they contain like large granules, multiple large granules in a very large nucleus. The lymphocytes and the monocytes, they do have blue or purple colored cytoplasm, and in lymphocyte, the nucleus is one large, which is occupying nearly completely all the cytoplasm. In the monocyte, the nucleus is kidney shape, and the cytoplasm is blue or purple. So, bit closer look for memory to remember these cells, which I think is important. And as a reminder, I believe we talk about this in our immune system lecture, and the different functions of T lymphocyte and B lymphocyte. T lymphocyte bind to the target cell, which is presenting antigen, and then T lymphocyte detach from that target cell, and finally the target cell will die by apoptosis. When it comes to B lymphocytes will change into plasma cell, which will secrete antibodies, and these antibodies will go and attach to the invading antigen, which is bacterium, and with the help of macrophage, these antigen antibody complex will be taken away. So I believe this is an important table, which is showing RBC, red blood cell, WBCs, or the white blood cells, and platelets. The lifespan here, if you look at that lifespan, LS, is for 100 to 120 days for RBC. There are no other cells which will live that long like RBC. And therefore, one important concept is in the bone marrow, like myeloid and erythroid ratio is 3:1. Like there are 3 times more myeloid cell than erythroid cell, which are RBC forming cells in the bone marrow. But in the blood, the result is opposite. There are more RBC and less WBC. How can this happen? I believe one of the answer would be like because of the longer lifespans of RBC. So now let's take a look at the platelets, which is also called thrombocytes. Platelets are not a complete cell. They do not have nucleus or organelles. They are just fragments from the much larger megakaryocytes, large bone marrow cells, and they are enclosed in the plasma membrane. Plate mean as a flat shape. They are like discoid shaped cytoplasmic fragments containing granules. They stay deep purple, and their diameter is really small, like 2 to 4 micrometers, so which might be like half the size of the RBC. And platelets, again, like they are also floating in the blood. And once there is an injury, then there will be exposure of the subendothelial factor, which is called and von Willebrand factor is deposited on subendothelial region, and that stimulate the platelet attrition on the capillary vessel. And there will be granulation released from the platelets, and then that will attract more platelets to come and join, and that is called the thrombus formation, primary platelet plaque formation. So I want to stop here for a while. I want you to take a break. I want you to go and look back the previous slides. So let's talk about clotting factors. Clotting factors are present in the plasma. If we remove the clotting factors from the plasma, we will get serum. So the clotting factors these diagrams show the coagulation sequence and how clotting factors work together to form a clot. First, activation turns clotting factor into enzyme. And once the clotting factor is activated, it activates the next sequence and so on and on and on in the cascade. And finally, we will get fibrin, which is insoluble polymer. And that way, that we reinforce the platelet plaque with the fibrin threads that act as a molecular glue for the aggregated platelets. So we have 12 clotting factors in our body, and I just wonder it might be worth studying this to some extent. So now we have a thrombus plaque, which is made of fibrin, and RBCs are trapped in the thrombus. So we always heard about red marrow and yellow marrow. What is red marrow? In the red marrow, we have immature RBCs. They make blood cells actively, and their sites are between the trabeculae of spongy bones. Yellow marrow, inside the marrow cavity, it is mainly filled with fat cells. And can I remind you that bone marrow, they are just reticular connective tissue. So these are the sites of the red bone marrow. So in between, as we say, like they're between the trabeculae of spongy bones, trabeculae in between the trabeculae of spongy bones here. So this is the micrograph of the red marrow. And this slide might be more important. So all the blood cells, they come from hematopoietic stem cells. But start from the very beginning, the hematopoietic stem cell will divide into myeloid stem cell and lymphoid stem cell. Lymphoid stem cell will develop into B lymphocyte and T lymphocyte. Myeloid stem cell will develop into pre-erythroblast, which is the precursor of red blood cell, myeloblast, which is the precursor of granulocytes, monoblast, which is one of the agranulocytes, and platelets. So the hematopoietic stem cell will develop into myeloid cell and lymphoid cell. Lymphoid cell will develop into lymphocyte, but myeloid cell will be responsible for RBC, most of the WBC, and platelets. So I think it is worth studying this diagram So from myeloid stem cell, we first become into pro-erythroblast. And pro-erythroblast we gradually develop, and then just before it became mature form, it is called reticulocyte. And finally, it will become erythrocyte. So as a closer look, hematopoietic stem cell, we develop into myeloid stem cell and lymphoid stem cell. Myeloid stem cell, we develop into RBC, most of the WBC and platelet. Just before the pro-erythroblast become RBC, red blood cell, the immature form is called reticulocyte. So now we will talk about sickle cell disease, which is caused by the abnormal hemoglobin of RBC. As we know, there are 4 globin chains in our RBC, and if there is any problem or issue with the amino acid components of this globin change, for example, like instead of glutamine, then it is replaced with valine, then we have this problem, which is called sickle cell disease. So there is abnormal globin change. And when there is a low oxygen environment, the RBC shape change from biconcave structure to a sickle shape structure. And therefore, this problem is called sickle cell disease. I'm going to stop the lecture here. I want you to go back and study. I hope this is not a difficult lecture.